Point is, I believe I have it labeled community or community structure or something like that. But as I was saying at the end of class last Monday, we really have been doing community ecology for a while. Okay, and so we just have to remember it's any type of interaction between organisms that are different species. And so, starting with our the intraspecific additions to our logistic growth model, we really started community ecology there. But we were still using population models, and so it kind of blurs the line a little bit. Today, we're going to get more specific into actual communities. You want to shut it completely, Katrina? Thank you. Okay. So we can take our communities and divide them into autotrophic, which auto should be familiar, like photoautotrophs. And so these are communities that are based on solar energy, such as forests and grasslands, in other words, plant ecology. Some of you are in plant ecology, right? So you can tell us, you might in plant ecology?
I took it.
You took it? Okay, y'all tell me if I'm telling you the truth about plant ecology. I took it a long time ago. I did take it though. We're in this class, we're really more interested in heterotrophic communities. And so chemoheterotrophs are animals. That's where the heterotroph is coming from. Heterotrophs are dependent upon the autotrophic communities, which is why you likely needed to take plant ecology if you're going to really understand how to manage for animals. I'm a woodpecker biologist by trade, and so I had to take dendrology, and in most of the projects where I was doing my research, I needed to know what kind of trees the woodpeckers were feeding on and stuff like that. And so it's really hard to understand the heterotrophic community if you don't understand something about the autotrophic community. So you just have to keep that in mind. On this slide, you get a specific guild, which is what we're going to talk about in a couple of slides. This is part of the bark foraging guild. And specifically, of course, all of these birds are woodpeckers, which are the best things in the whole world. As ecologists, it's really hard to study if you want to be really true community ecology. In other words, if you really want to study all of the organisms interacting in a particular place, that is a big task, right? Because most of us don't have that type of expertise. I don't. I can do the birds really well. If you press me, I can do some mammal stuff. I have had ick, but it's been a while. And so it'd be really hard for me to get in there and to really put together a true community, because I didn't even mention all the plants that we need to talk about, or the microbes, or anything like that. And so more often than not, we refer to it in a more narrow sense. And then sometimes we'll put them together. And we're trying to do that more and more into larger and larger, more encompassing models, ecosystem models. I guess 2 weeks ago now, I went to the Tennessee Academy of Sciences meeting, and I didn't think I'd be that interested in our plenary speaker, the big speaker we all have to listen to, or we all listen to, I guess. But he was talking about the Cray supercomputers over at Oak Ridge, and it was actually really neat. But one of the things that the newest Cray, and I can't remember all the specs on it, he gave us all that, of course, how fast it is, but they're trying to do more ecosystem level modeling and putting in as much information as possible into the supercomputer to see what comes out. That's becoming more and more important, and then coupling that to their weather models, and their weather models can predict out several months instead of a few days like our local weather and stuff like that. It's really interesting. Anywho, in order to build those models, any of those models going into the Cray supercomputers, you need people with different types of expertise, right? So just looking at this for the fish, bird, and mammal community, you're probably looking at 3 different people if you really want to do a good job at it, because we tend to be specialized in that way, okay? The botanists, this is where the plant ecology people can chime in. But the botanists, they tend to use the word association a lot of times when they're talking about communities, like moist soil plant associations and things like that, which also refers to finding groups of different species together in certain locations. So we have a course offered every once in a while called Aquatic and Wetland Plants. And one of the things we did in that class, although I didn't take it here, I took it somewhere else, was wetland delineation plants, for example. So plants you would expect to see in an area regardless of whether it's flooded or not when you sample that area. That's actually a very important skill to have. It's a marketable skill. You can get a job doing that type of thing. All right. Now, before I got even more specific than bird community, because even knowing all the different bird communities can be a problem, and so a lot of times we'll break them up into guilds. And so guilds carry out similar functions. In other words, they have similar niches in the environment, even though they may not be taxonomically related. Here, of course, on the pictures, I gave you my favorite guild, and now I've got more specific. It has a woodpecker in the middle, a cute little downy woodpecker. But it has 3 other members of the bark foraging guild. The black and white warbler, which is a really cool warbler, passes through here mostly on migration, will circle around the trunk and eat off the vertical tree trunk just like a woodpecker. Doesn't use its tail as a prop, so it looks a little weird. The nuthatches, so there's 4 species of nuthatches in North America. This is the most common around here, white-breasted nuthatch. They are pretty distinct because they can run down the tree head first. Woodpeckers can't do that. So they're pretty easy to pick them out. Fun to watch at the bird feeders. And then the other one I have up here is a brown creeper. And brown creepers do move a lot like a woodpecker. You can see in the picture they use their tail as a prop. But woodpeckers can hitch up and they can hitch down the tree. Brown creepers typically will only go up, and they tend to spiral. And so they'll spiral up the tree, go to the base of the next tree, and spiral up the tree, and fly to the base of the next tree, and so forth. But all of them are using the vertical tree trunk. So they are part of the bark foraging guild, even though none of them are that closely related to one another. Probably one you may have encountered before if you're more into aquatic biology is some of the macroinvertebrate surveys that they do. Instead of worrying about getting those species down to species, because that can be very difficult, especially for some of the groups of macroinvertebrates without being a little bit more specialized than most of your technicians are. They'll group them as to whether they're filter feeders, predators, scrapers, and things like that, and then they will compare the ratio of those different groups to one another to assess the health of the stream. So this idea of guilds can be very important, depending on what you're doing. But again, what we're really doing is saying our expertise are very difficult for us to grasp beyond, on an ecosystem level, would be considered a very small unit, right? So if I go in and I'm studying the bark foraging guild, that's a lot for me to accomplish as a single individual. But that's only one guild of birds within that forest. It certainly doesn't tell you what is happening within the whole community. Right? Community ecology can be difficult in that way. Much less ecosystem ecology, which I think you get a little bit of in plant ecology, but not so much in here. So ecosystem biosphere level, ecosystem level is where you do your nutrient cycling, large-scale nutrient cycling and energy flow and stuff like that. And you start to incorporate weather patterns. Not so good at that myself since I'm at this level mostly. All right, so when looking at communities, we tend to look at structure. That could be the biological structure or the physical structure. And so the physical structure is going to be like the topography, the soil type, the soil pH, things like that. So kind of the abiotic part for the most part that actually forms where the organisms are living. So for us, the physical structure would be like our bridges and houses and things like that. That would be our physical structure. Biological structure is created by the organisms themselves, and it represents the pictures that I have on this slide. And so on this side, this is when I visited the Redwoods National Forest, which was super awesome. It was also when I learned that sequoias and redwoods were not the same thing. Which I should have known a long time ago, but sometimes you just have to see things and experience them yourself for them to sink in. And so you can see a little scale there. That's me on the side of the tree for scale. Big old giant ferns in the foreground. And so those trees are going to create layers. And those layers are going to be used by different types of species. So it's almost like an apartment building, but it's alive. And so that's biological structure. And then on one of my treks out west, I went by the Grand Prismatic Spring. You might have been to Yellowstone? That's at Yellowstone, Grand Prismatic Spring. In the center, it's got water that comes up, the hot spring part, and that's like a deep blue, really pretty deep blue color. And then as you move out from there, you start getting yellows and reds and oranges and browns. And those colors are created by bacterial mats. And so this is just a picture of one of the bacterial mats. But what caught my attention was there was one of those little informational plaques as you walked along a boardwalk, because you don't want to get down into the water for lots of reasons. And it explained that these mats have different layers made up of different species, in this case of thermophile bacteria that live in hot conditions, and that there are even things that live in between and associated with those different layers within these mats of bacteria. And so the layering, the biological structure that we're talking about here, can be at a microscopic level or a macroscopic level. So I chose my pictures on here because of that. I sometimes work with Dr. Hoosman, microbiologist, immunologist, and we were working on some bacteria that live in the gut of yellow-bellied sapsuckers to see if they could do cellulose degradation. Specifically, could they use cellulose as a food source during the winter? And I was reading some papers trying to get up to snuff because I'm not a microbiologist over here, never had microbiology. And I was really interested because the paper was written by a microbiologist who very obviously had not had very much ecology. But what they were describing was the microbial community, and specifically some mutualistic associations where you have some bacteria within your gut that harvest oxygen. And in the process of harvesting and using oxygen, it ensures the anaerobic environment that other bacteria in your gut have to have. That oxygen is toxic for them. So I thought that was really cool. So you could study biological structures in all different types of places. All right, so we're going to focus a little bit here on biological structure. And we're going to start with some stuff you're likely somewhat familiar with. We're going to start with the idea of dominant species. Dominance can also be associated, or in your mind, you can think influence if you want to. But basically, these are species that shape what the ecosystem is going to look like, what the community is going to look like. We're going to talk more about that on the next slide. And then likely in freshman biology or somewhere along the way, you've been asked to calculate species diversity. And so we are going to talk about species diversity as well. If you've had me before, we're going to use the same old species diversity slide. And we're going to talk about how we incorporate species richness, which is the number of species, and species evenness, which is how abundant is one species compared to another. But we are going to get more specific, which you may or may not have done in other classes, and we're going to talk about alpha diversity versus beta diversity and gamma diversity. This picture is from Yellowstone as well. You're probably aware, because I think at least in some of the NRM classes, you're going to talk about the big fire at Yellowstone and why it burns so deeply and why so many trees died and stuff, probably at some point. Fire climax systems. Y'all aren't looking at me like you did too much. Okay. Well. So Yellowstone had a big giant fire. And it killed a ton of forest because they had not had any type of leaf litter control. It's before we knew much about controlled burning. And so usually you learn about it into your introduction into controlled burning and why it's important in some of these ecosystems. And so if you don't burn regularly or have some type of litter control regularly, then it can accumulate. And when you have a fire, it can burn too hot. And even trees that are adapted to a fire climax system, which means they have like squishier, thicker bark on the outside of them, can be killed, which is what happened at Yellowstone. It burned hot, and it killed a lot of habitat. And so this picture always reminds me of that because they were growing in a meadow in one of the areas that still had scorched trunks standing, left over from the big Yellowstone fire. Okay. So let's talk about dominance first. So just on this one slide, talk about it a little bit. Apparently, I had just been to Yellowstone when I made these, because this is Yellowstone too. If you haven't been, you should totally go. It's awesome. But be prepared because it's just got one loop, and then you can pull off in different places along the loop and do different things. And if you pull off, it's not so bad. But the loop gets backed up, traffic forever. It's kind of crazy because you go into Wyoming and all this and you don't see a soul. And then you get to Yellowstone, it's like New York City. It's kind of weird. But it is a remarkable place. And when we were there was a fire on one side, so we all got redirected. Everybody got redirected in the same direction, so it got kind of messy. So we went to the Tetons, which are just 20 minutes from Yellowstone. Highly recommend. Wasn't on our list, but we went anyway, and it was awesome. Not quite as many people as Yellowstone. All right, so dominance can be a variety of different things. And so if we were in a grassland, grasses are going to be the most numerous plant there, and certainly they're going to be driving the ecosystem. Grasses have fibrous root systems which intertwine with each other and lock the soil in place and accumulate organic matter and things like that, which is going to be super important for nutrient cycling. And I think a lot of times you jump to most numerous kind of off the bat. But it doesn't necessarily have to be most numerous. For example, if we were looking at a forest like you see here, if we counted up all the plants in the forest, trees wouldn't necessarily be most numerous. Depending on the canopy cover, it could be herbaceous plants or ferns or something like that on the forest floor. But because they possess the most biomass, they certainly have the biggest influence on nutrient cycling. And then because they make up the canopy of the forest, they control how much light is able to penetrate and what grows underneath. And so it could be the thing that has the most biomass. It could be what takes up the most space, makes the largest contribution to energy flow, or does something critical in terms of nutrient cycling. And the last one I always find kind of curious, so we'll use an animal example for that. And that is their presence influences the rest of the community in some way. And so I think way back, early in the semester, when I lectured for like days and days about niches, it wasn't that long, but it might have felt like it, we talked about the barnacles, Thamylus and Balanus. And we said that the Thamylus lives at the upper part of the rocks, and the Balanus lives on the lower part of the rocks, and the Balanus are always underwater. They don't seem to tolerate low tide very well. Thamylus can live in both of those environments, but gets pushed to the upper environment because of competition with Balanus, which is a larger species. Does that ring a bell? All right, well, what we probably didn't talk about, I don't remember talking about it at that point, is that is a simplification of that particular community, because there's also another thing you can add into your experiment, and that is a barnacle-eating snail. So we used to do a model on the computer, and we'd add the snail to see what would happen. Well, it turns out the snail prefers to eat the Balanus, which is the larger of the two, which might be partly why it likes it. You get more bang for your buck if you can take it and eat it, right, than eating the smaller Thamylus. But they'll go in and they'll clear out spots on the rocks where they're eating the thing, killing the Balanus, and that creates openings that allow the Thamylus to come down and colonize those areas, increasing the diversity below the waterline, so that you have both species rather than just one. And so the presence of that predator is shaping the way that particular community looks. Right? So that's what this is talking about. And there's lots of examples of how things like that can occur. All right, any questions about dominance? So it's kind of interesting. We don't spend a lot of time on it in this class. I don't know if you do plant ecology because I didn't have Dr. Kruger. But when I teach the tree sampling, we talk a lot about the trees in particular because they do have such a big influence. And so whichever trees are having the greatest, in this case, assign an importance value to them, so the one that's most important probably has a lot of control over light and nutrient cycling in particular. So, a bit of a concept that I at least have a grasp on from plant ecology. Okay, let's talk about diversity. All right, so we're going to get more specific. The diversity, when you've calculated probably the Shannon-Wiener or Shannon index, maybe a Simpson's index. I was pretty pleased. I like the Simpson's index more than Shannon-Wiener for a few reasons. But you see Shannon a lot more, and I was pretty pleased when I was at the meeting. Somebody was going on and did all their calculations with Simpsons, and I was like, yeah, I'm not the only Simpsons person out there. But those types of indices that you calculated, like in freshman biology, if you took biology 130 here, you did the Shannon index. That's alpha diversity. Okay, so we're going to be more specific. It's alpha diversity. That is diversity within a single ecological community. Now, what we usually do is we'll do alpha diversity of this community, and then we'll calculate alpha diversity of another community, and we compare them. That's really getting you more to beta diversity when you make the comparison. Okay, so just kind of keep that in mind. The freshman lab here do insects in a frequently mowed field and an infrequently mowed field, I believe. And so you calculate the diversity for this, the alpha diversity for this field, and the alpha diversity for this field, and you can compare those numbers. Okay? We're not to that comparison yet. So alpha diversity is within a single ecological community. And then the second thing on this slide is just reminding you of what the definition of community is. So all the populations that are in that area. But keeping in mind, even in the example I just gave you, we tend to look at a subset of the community. In that case, the insect community. All right. So alpha diversity is normally described by the species richness and the species evenness. And so there are really two terms when I'm talking to my freshman biology to try to separate out. There's kind of like biological diversity or biodiversity, and they are the same thing, but biodiversity is often just used to describe the species that are present, and that's just one part of the actual calculation, right? And so when we're talking about alpha diversity, and like I said, it really is biodiversity as well, what we're really discussing is a way of measuring that diversity. How do we measure what's there, what's present? And so as we talk about species richness and evenness, what we're talking about are two parameters that we're going to use to build a model. Okay? In this case, we're building diversity indices. Okay? And so if you picked up the ecological methodology or the ecological statistics books in my office and read them, woo, they're exciting. Neither one of them are very exciting. We had to read the ecological methods one when I took graduate school for field methods. But there's a whole chapter just about diversity indices, what I'm trying to get you to think about here. And each diversity index is usually named after whoever put it together, like Shannon and Simpson, to give you examples. And each one is going to weight these parameters differently. Okay? And so the parameters are probably familiar with you. The first one is species richness. That's the easiest thing to collect. Whether the species is present or absent is all you really need. And so if you're interested in birds and bird watching, you can go by the state park or the wildlife refuge headquarters, and they will have a checklist of species for that area, which is very handy. It's a very good resource. And then you can ride around and hike, and you can check them off that list. That is important information, but all it's really going to give you is species richness, what's there. Okay, we call that type of information presence-absence data. And it is very important, and sometimes we're going to weigh that heavier than maybe other times. Okay? Species evenness, on the other hand, is the relative abundance of one species compared to another. That's the relative part, right? And so is this species common or is this species rare? Which we talked about extensively in ComBio. We also talked about rarity, which we won't get into in this class. And how do you calculate rarity and what goes into determining whether a species is rare. Okay, that's very important. But that comes out of the species evenness. And so you put these two together, I'm going to go on to the next slide because it's got the same stuff basically on it. And you build your index based on those two things. And so let's start generally. And so one of these days I'm going to change it because I have had a couple of colorblind students say that they can't tell the differences here. That's fair enough. So I need to get one that doesn't have colors. But if you look at the top, our species richness is going to be represented by the different colors representing different species, essentially. So we've got red, orange, green, blue, and purple. And so species richness in the first community is 5. And then down here, if you look carefully, you also have orange, red, blue, purple, and green. So a species richness of 5. And so if we stop there, then we would say that they're equally diverse. And so what they're trying to illustrate is the importance of measuring more than presence-absence data. It's a good start. It tells you what's there. And as we move into beta diversity on the next slide, it can allow us to compare two different sites to see what's missing, for example, or what's present, what they share, which is very important. But it doesn't tell us a whole lot or delineate between these two communities. And so if we add in evenness, or how common one species is to another species, then the one at the top is much more even. In other words, you have equal numbers, approximately equal numbers of each color of dot. And the one at the bottom is uneven because we have lots of green dots and very few of the other dots. Now, this is the way we generally teach alpha diversity. And I feel like it gives you the impression that this is good up here at the top, all right? Because that is the way our indices work. If we did a Shannon's index, it would say this one is more diverse than this one. If we did a Simpson's index, it would also tell you this community is more diverse than this one. However, that's not really reality. In reality, most communities look like this one. All right? And so keeping in mind that what we would be doing is comparing communities that all look like this, and that's where the indices come in handy, because what it's going to tell you is if a community is missing something and you can look more closely at it. Now, before we miss this idea, I want to, again, kind of go back and talk about the two indices I keep mentioning. We're not going to worry. There's dozens that you can choose from. And nowadays, you can also do some modeling. So, for example, if I wanted to really do species richness, if you listen to some of Dr. Blanchard's students, they do curves. And what you do is you go to your site, and at first you're going to be finding lots of new species because you've never sampled there before, right? So every species is a new species. And then over time, that curve's going to flatten off. In other words, your odds of finding a species you haven't encountered before is low. Not impossible, but low. And so that's generally what you do for species richness. And so then you can compare those curves to one another. So we did that with woodpeckers in a Tupelo swamp versus a mixed hardwood swamp. And we found that the woodpecker curve in the Tupelo swamp flattened out here, and then the curve for the mixed hardwood forest flattened out higher. Okay? And then we were able to put confidence intervals on them and show that they didn't overlap, and so we had higher species richness in one community than the other. We definitely do that type of work. And that gets pretty complicated to build that model because we use something called bootstrapping in order to build our confidence intervals. But mostly what we do is we do incorporate the relative abundance. And so if you consider the Shannon index that you, if you went to school here for Biology 130, you calculated. If you had general ecology here, you also calculated in there. It doesn't have a scale per se. What it'll say is it varies from 0 to higher. And it says in most ecological systems it doesn't go above 5. And so it's kind of 0 to 5, sort of. Most of the time we get 1, maybe 2. But you don't have an upper limit. So it's hard to say whether diversity is high or low. And then Simpson's index, which I like to use because you can get some idea about diversity. It varies on a scale of 0 to 1, where 0 is low diversity and 1 is high diversity. And so if I get a 0.8, then I know that I'm doing pretty good, that there's pretty good diversity in that particular habitat. All right, so that's one of the reasons I like Simpson's. Now, what I didn't tell you about the Shannon scale, the one that's 0 to infinity potentially, it tends to increase with species richness. In other words, it incorporates species evenness, but it weights species richness higher. Whereas in the Simpson's index, it's the reverse. It weights evenness higher than it weights richness. And I'm pointing this out because that's what you have to decide when you're choosing what type of diversity index you want to use. Is which of these parameters is more important for the question that you're asking, and what kind of information do you need? If you're just sampling one community, doing a Shannon index isn't going to tell you very much because you don't know what you're comparing it to. Whereas if you're measuring in one community, you can do a Simpson's index and you can at least say, all right, it varies from 0 to 1, and I know where it is on that scale. That makes sense? So there's a lot to consider. If you're really interested in it, like I said, I've got a whole chapter in my office you can read. It's very boring. All right, any questions about that before we do one more thing before we do off the beta diversity, I guess. Any questions about that so far? Okay, the last thing I want to mention is what I started a second ago, and that is when you look at this community, like I said, this is going to be more normal. So for example, if I'm interested in the mammal community in the bottomland hardwood forest in northwest Tennessee, this would be mice let's see, looks like the red is the next. That would be our white-tailed deer maybe, because they're larger, they take up more space. And then this blue dot over here, maybe that's a bobcat, right? And so if you're thinking about the lifestyles of those species that I just talked about, that makes sense. You would expect to have more mice, fewer deer than mice, fewer bobcats than deer, right? That kind of thing. If I'm looking at the mouse community, then maybe these are deer mice, these are white-footed mice, and this is some little jumping mouse or something like that. And then what I need to know is how common do I think that jumping mouse should be? Is this a rare species or is this a common species? Is this something I would expect to have as many spots as the green species, or would I expect to be something I wouldn't encounter very often? And if it's something I wouldn't encounter very often, then you need to look at what's causing it. What's causing the species to be rare, or to use our terminology in the United States, endangered, right, or threatened. All right, I think that's the last thing I want to say about that. Okay, let's move on to beta diversity. And so now what we're going to do is actually compare communities to one another. Okay, and it's really hard to talk about alpha diversity without stepping on this one a little bit, because when I compare the diversity between a frequently mowed field and an infrequently mowed field, then really I'm stepping up to beta diversity, right? I'm comparing between two different communities. That's what it is by definition. I don't think, let's see, no, I don't think I talk about it in here. I'm just making sure I don't. Nope. Another thing that we commonly do is we measure what's called similarity. You may have run into that before. And so similarity looks to see what kind of species two different habitats have in common. That's also beta diversity, and it can be very helpful. And so if I take this community that we had before with all the dots on it, and I compare it to another community, I can see which species are missing and which species are present. Like what's making up this community compared to another one. That's also beta diversity. We're going to focus on the larger scale beta diversity concept. And so it gives your changes in regional diversity, basically. And so it's not necessarily trying to do this community versus this community, but rather what it's trying to do is what happens as you move from this community to this community. What species are lost and what species are gained, essentially. And so typically this would be something like if you want to do a mountain study, and you started sampling plants or animals at lower elevations, and then you sample every 1,000 feet or whatever makes sense going up the mountains, and you look to see what elevation you lose this species, what elevation do you gain this species. That is beta diversity. All right, if you're doing fishes, maybe you want to do fresh water into salt water, because that creates a gradient, like where the Mississippi River meets the Gulf of Mexico. And so you would start up in pure fresh water, work your way through the salinity gradient to pure salt water, and see when species are lost and when species are gained. Okay, that's basically what beta diversity is doing. And so this is measuring the rate of change along a gradient. And on the next one, I have a picture, and it starts our discussion saying that beta diversity allows us to do three very important things. We can measure changes in diversity as you have changes in environmental gradients or environmental factors. I stuck with birds or animals here rather than plants. Plants are often used in this example, right, as you're changing elevation. And so I've got birds instead. One of the travel studies we did a few years ago, we got to go to the cloud forest in Ecuador. And we went bird watching, and we saw all kinds of cool things, like toucanettes, which are little toucans, and squirrel cuckoos and stuff like that. Little black and white owl, which was really pretty. But we stayed at a sanctuary, a birding sanctuary, the Milpy Birding Sanctuary near Mendo, and I don't remember the elevation there. And then they had a second site, which was I think only 500 meters. No, kilometers, probably 500 kilometers difference. So no, that's too far. It was remarkably close, so maybe like 100 kilometers. I think it was within an hour's drive. But it went that much higher up the mountain. It wasn't that much higher up the mountain, is my point. And the bird communities were completely different. You could get your checklist and then move up and then do a new checklist and really build your life list, essentially. It was pretty awesome. And so they can be quite sensitive to changes in elevation, as you can see here. Well, if you look in between them, you get 1,200 meters. The next one's at 2,000 meters. So it might have been right on with the first thing I said that popped into my brain. And so it's really cool to look at different things along a gradient. But the second thing it can do is whether the species are sensitive or insensitive to change. Get all up there so I don't get lost again. Okay. In other words, as you're going up the mountain, just stay with that example. If we're going up the mountain, do you see this species at several different elevations before it falls out? That would be relatively insensitive. Or do you see it at a very particular elevation and nowhere else? And so that's going to be very sensitive. That's what it's talking about. So if we're doing the salinity, for example, are they freshwater only? Or are they saltwater only? That's going to be relatively sensitive, and there are a lot of fish that are like that. Or do you find them at a variety of salinities? That would be insensitive, right? That they are good at living anywhere between freshwater and saltwater. All right, so once we can figure out which species are sensitive and insensitive, what that allows us to do is to figure out if our communities are interdependent or independent. Interdependent means that the species that make up that community persist because of the presence of the other species. Whereas independent means that they just grow where the environment is proper for them, and then they don't grow where the environment is not proper to them, and it doesn't matter what's around them. Now, like all things in ecology, life overcomes, right? And so the real answer is probably somewhere in between that. Because just because you find plants living together or animals living together in a community, and you don't find them outside that community, doesn't mean that they're necessarily interdependent. It just means that they all like to live at that elevation for whatever reason, right? So it's hard to prove that they're actually interdependent without doing some removal types of studies. So you kind of have to keep that in mind. But there are definitely assemblages that have meaning. So earlier today I mentioned wetlands delineation, that there are groups of wetland plants that if they're present tell you that that land is flooded at least for part of the year. And those tend to be the same plants no matter where you go and look. The question is, does that mean that they need each other to survive, or does it mean they need moist soil to survive, right? And so it becomes a little bit fuzzy in that regard. Now, when you're doing these types of things, for independent, that's going to be the species that you see growing at times where they're not with the other species in that community. That's pretty easy to kind of filter out. Interdependent's a little bit harder to filter out. Do they really need each other? All right, any questions about that one? All right, so the first one got our gradient. You know, what are we losing and gaining along the gradient? Now we're looking at the communities themselves. Are these plants or animals that make up the community dependent upon each other, or do they just live where the gradient is appropriate? For the last one of the things it allows us to do, because we're looking at species being gained and lost, is we can either look at different points in time. In other words, the idea of succession, which I'm sure you talked about in plant ecology. And so succession is how communities change over time.
Thank you.
Plants are, again, normally the textbook example here, but when I'm teaching freshman bio, I use birds. So you have birds that live in grasslands, and then you have birds that live in edges, and birds that live in forest. To be fair, I show the plant changing. As the plant community changes, the bird community changes, right? Probably my favorite example of succession was, again, a microbiologist. We had a microbiologist that's no longer here. Her name was Dr. Liu, and she gave a presentation about fermentation of sauerkraut. And what she did is she showed the bacterial community, and as it changed, in different stages of the fermentation process. And it was the same as what I was telling you earlier. She wasn't using the terminology we would use to describe succession, but that's what she was describing, right? How these species are changing as the environmental conditions change. So I thought that was really cool. Not only can we do things, measure successional changes in a community, what's happening over time, we can also look at the effect of disturbances. Those can be natural disturbances, such as a tornado comes through and rips up the forest. What happens? What comes back? How long does it take? How do you fill it in? You know, what species grow first? All that kind of stuff, just like succession. Or it could be man-made disturbances. So we've been farming this land in our family for 30 years. Now we want to let this field go fallow. What happens from there? That kind of thing. Or if we continue to disturb it, what happens? So that's pretty handy thing to be able to do. Any questions about beta diversity? So it can be through time, it can be through disturbance, it could also be along a gradient. There's a lot of ways that we can use beta diversity. But basically it's trying to figure out what's where in the environment. All right, and so that leads us to gamma diversity. In gamma diversity, this is where species, we're trying to figure out what is the diversity of species across larger landscape scales. This is where we're actually going to do more of your community delineation. It really is going to lead more into ecosystem delineation, although to get to ecosystem level, you've got to put those abiotic factors in there, which are going to be driving where the communities are. It's hard to talk about one without the other in gamma diversity. And so in more measurable terms, this is my favorite, although it's kind of hard to remember, but it's the product of the alpha diversity, so what do you have at this specific location, and the beta differentiation, in other words, how we're changing between those different communities that we're sampling. And what that's going to allow you to do is to say, all right, here is one community, and the alpha diversity of this community. Here's a completely different community, and the alpha diversity of this community. Okay, so that's still alpha diversity. Beta diversity is then going to look at how do you get from this community to this community? What are the changes that happen along the way? What is the gradient that's present that caused that change? And then gamma diversity is basically going to circle those and say this is one particular community and this is a different particular community. And then do that across a larger landscape. So as you're getting into gamma diversity, this is going to get more into some larger scale modeling. And then some of you may have had some courses in like GIS and stuff like that where you get to adding different layers to see how things are going to shake out. That's getting into this level of diversity. Quite far from the Shannon diversity we start out with freshman biology. But really cool. And so I've got a little bit more. We're going to talk about physical structure on the next slide a little bit. So all this has been about biological structure. But what we're not going to get to today is the island biogeography. And then very briefly, just island biogeography has been replaced by metapopulation model, which we've already discussed. And then how to kind of use metapopulation model at a community level. Because by definition, a metapopulation model, which you're supposed to know about already, does one species at a time. And so how do we take the metapopulation model for this species and then put another species metapopulation model on top of it and another metapopulation model on top of it? And as you do that, you begin to be able to delineate different communities. Right? So that level of research is gamma diversity for the most part. All right, did that make sense? Do you have any questions about gamma diversity? Okay. So your physical structure it was a little simple at first because we've been building quite a lot. We're doing a lot of community ecology today. Is that abiotic factors, including things like topography and stuff that I mentioned before. But the physical structure can also be made up of that biological structure we've been talking about. That could be what gives you the actual physical structure that organisms are using as their highways, as their apartments, right? Think squirrels in a forest. They got a little cavity, that's going to be their apartment, and then they run along the tree branches. The tree branches are acting like their highways, right? That's their infrastructure, if you will. We can break down that biotic infrastructure into vertical and horizontal layering. And so that's why I have a picture of the forest there, because it has very good vertical layering. And so as we look at a forest, we have the canopy. The canopy is going to be made up of the big, large, huge biomass species. They're going to be influencing how much light is passing through, so how much photosynthesis can take place underneath the canopy, which is going to influence whether or not you have some of these other layers even. But let's say we've got a fairly open canopy, so then you have an understory, which is going to be your smaller trees, those shade-tolerant trees. Then you get the shrub layer, maybe a herbaceous layer, down to the forest floor, and then some of you are NRM, so you've probably had some soil science, and so you get your layers in the soil, like the O layer, which is the organic matter, and A's and B's, and I don't remember what any of that means anymore. I did take it a long time ago, but it's been a while. And so all of those layers represent different places and different niches that organisms can take advantage of. And so as you increase vertical layering in an ecosystem, you're going to increase your diversity. And so it's not really a fair comparison, but if you compare the diversity of a forest, let's say birds, to a grassland, a forest is going to have a higher diversity because it's got more niches and more available layers for them to take advantage of. It's a little bit more fair to compare different forests to each other and different grasslands to each other, but it's not going to be as dramatic a difference and harder to show the differences in layering. Similarly, we can look at horizontal layering. And so to get this picture, I like this picture, but I can't find a good picture of just straight up horizontal layering. And so I took a succession picture and took the timeline off. That's all I did to get this, all right? And so all you have to do is walk across the landscape, and as the landscape changes, that's your horizontal layering. So you have a grassland layer, a shrub layer, an edge layer, and then getting down into the forest. Those are the different layers as you go across the landscape. This could reflect things like disturbance again. I don't think I have disturbance on my list. Oh yeah, I do. It could be a disturbance. It could be changes in the soil pH or chemistry. It could be a change in topography if you're going from high elevation to low elevation. That was probably one of my favorite labs we did in dendrology. We started at the base of the hill and we looked at the types of trees, and then we worked our way up to the top of the hill, and we saw the tree community change from the wetter area to the drier area. That was pretty cool. It could reflect a large disturbance or just as simple as a large canopy tree fell, creating a gap, and then what grows in, it takes advantage of that light that's now hitting the forest floor. Now, as you might expect, as you increase, or what are we doing now? Yeah, horizontal layering, you're also going to increase diversity. Which leads us to what happens when two communities come together. It's going to create something called an edge, and that tends to affect diversity as well. And so we're going to do transition zones, biogeography. We're going to review very briefly metapopulation, and then look at a couple of ways to build models, very briefly, on Wednesday. We're out of time. We got to exactly where I wanted to get, though.